The mouse we are talking about today borrows that same credibility, a golden, 3 times longer, curly-furred lab mouse that Colossal unveiled in March last year and called a step toward bringing back the woolly mammoth. Here are 15 facts about that mouse, and the math at the center of it is not the one the press release led with. The announcement landed on March 4, 2025, backed by a preprint on bioRxiv. A preprint means it had not yet gone through peer review. It still is not. The paper was revised as recently as October 30, 2025, and Colossal has said it plans to add cold tolerance and behavioral data before submitting it to an actual journal. None of that slowed down the coverage. Outlets from TechCrunch to CNN to NPR ran it within hours, most with the Mammoth connection in the headline and the preprint status buried several paragraphs down, if it appeared at all. That gap matters more than it sounds. Peer review is the step where independent scientists in the same field check a paper's methods and conclusions before a journal will publish it, and it is the difference between a company's own account of its results and an outside check on whether those results hold up. A preprint has none of that yet. It can still be accurate. It has simply not been tested by anyone with no stake in the outcome, which is exactly the group best positioned to catch an overstated claim. Plenty of preprints hold up fine once reviewed. The point is not that this one will fail. The point is that well over a year after the announcement, the check that would tell us either way still has not happened. The mouse itself carries 8 gene edits spread across 7 genes. One gene received 2 separate edits, though public reporting has never clarified which one. Stacking 8 changes into a single embryo and getting a healthy adult out the other side is a real engineering feat on its own, independent of anything to do with mammoths. Every additional edit raises the odds that something goes wrong before birth, an off-target cut, a lethal combination, an embryo that never makes it to term. Geneticists outside Colossal have said as much. The achievement is real, whatever else turns out to be true about the mammoth framing. Stack enough edits into one embryo and the failure modes compound. One edit can land cleanly, and a second can disrupt a gene sitting near it on the same chromosome. A third can trigger an immune reaction the first 2 never touched. Most embryos carrying that many simultaneous changes do not survive to birth at all, and the ones that do often carry defects nobody was editing for. A live, apparently healthy adult mouse carrying all 8 edits intact is the actual headline result here, if you are grading the engineering itself rather than the marketing built on top of it. There is a reason this work happened in a mouse instead of an elephant. A mouse goes from a fertilized egg to a full adult in weeks. An elephant takes 22 months to gestate, and the Asian elephant, the species Colossal is actually editing toward a mammoth, is itself endangered. Nobody gets to run 8 experimental edits on an endangered animal to see what happens. A mouse lets you learn whether an edit is even survivable before anyone risks it in a species that takes 2 years to tell you if you were wrong. Now walk through what those 8 edits actually do, because this is where the story gets specific. FGF5 controls when a hair follicle stops growing. Turning it off is why the mouse's fur grew roughly 3 times longer than a normal lab mouse's. MC1R governs the dark, agouti brown coloring most lab mice carry. Editing it is what turned this mouse gold instead of brown, a change geneticists have understood for decades. FABP2 controls fat absorption and metabolism, and Colossal engineered a version that mimics a trait actually found in ancient mammoth DNA, tied to the thick subcutaneous fat layers that helped real mammoths survive the cold. That one is aimed at physiology, not just appearance. KRT27 is the specific one worth sitting with. It carries a real mammoth mutation, a single amino acid swap at position 191, methionine to valine, pulled directly from sequenced mammoth genomes. TGFA was edited alongside it to help produce the coat's wavy texture, and it is tied to the same kind of mammoth-specific sequence. Between those 2 and FABP2, that is 3 edits genuinely traced to something a mammoth actually carried. The remaining 3, FAM83G, FZD6, and TGM3, all shape hair follicle structure. And they were chosen because editing them reliably produces curly, woolly-looking fur in mice generally. Not because a mammoth's version of those genes has been confirmed to differ the same way. It took 3 separate genes just to get the curl and thickness looking right, which tells you how many individual dials sit behind something as simple sounding as fur texture, and how much of this project is tuning an appearance rather than transplanting a mammoth switch. Add it up, and the ratio is 3 to 5. 3 edits: TGFA, KRT27, and FABP2, traced to real sequenced mammoth mutations. 5 were picked because their effect in a mouse was already well documented, then aimed at a mammoth-shaped result. That distinction, a gene with a confirmed ancient mammoth variant versus a gene chosen because editing it produces a familiar look, is the entire gap between reconstructing an extinct trait and imitating one. Both are genuine science. Only one of them is actually mammoth biology. Colossal says the underlying targets came from comparing 59 ancient mammoth genomes, ranging from 3,500 to 1.2 million years old, against living elephants. That database is real and genuinely large, spanning almost the entire span mammoths existed as a species, which makes it stranger that only 3 of the 8 edits trace back to anything found in it. The comparison existed. Most of the edits did not come from it. Here is what has not been tested yet either: whether any of this actually helps the mouse survive cold. The preprint did not include cold tolerance trials, and Colossal itself has said that data is still to come before the paper goes anywhere near a journal. So the mouse has golden, 3 times longer, wavier fur and altered fat metabolism. And as of right now, nobody has published a test of whether any of that keeps it warmer than an ordinary mouse in the cold. The look arrived first. Proof it does anything useful has not. A real cold tolerance trial would mean measuring core body temperature and metabolic rate in edited mice against ordinary ones at low temperatures over time. The kind of data Colossal has already said is coming before the paper goes to a journal. Until that data exists, the fur is a hypothesis wearing a finished coat. 4 scientists outside Colossal responded to the announcement, and it is worth hearing them by name rather than as a blur of critics say. Tori Harridge, a paleobiologist at the University of Sheffield, said a mammoth is much more than an elephant in a fur coat, and that this approach was never going to bring one back. She was not weighing in on the engineering. She was pointing at the label on the box. Louise Johnson, a geneticist at the University of Reading, went further, noting that most of the edited mutations were not mammoth-specific at all, just genes already known to do the job, and that the result would not be a mammoth in any meaningful sense. Her point was not that the science was fake. Picking a gene because its effect is already documented, rather than because a mammoth is known to have carried that version, is a shortcut, and the headline never uses that word. Jacqueline Gill, a paleoecologist at the University of Maine, raised a different problem. A gene switched off in a mouse can behave differently once switched off in an elephant, an animal with a different body, a different metabolism, and a lifespan 40 times longer to develop side effects in. A lab mouse lives 2 or 3 years. An elephant lives 60 or 70. A subtle problem caused by an edited gene could take decades to surface, long after the mouse carrying the same edit had already lived out its full life looking fine. Vincent Lynch at the University at Buffalo called the project exactly what its preprint status suggests it is: a proof of concept, showing that several edits can be stacked into one animal without killing it. Nothing more claimed, nothing more proven. Lynch has said versions of this about several Colossal projects, which is worth knowing before weighing any single announcement from the company. He has made a similar point about the company's dire wolf project, where the resulting animals carry 20 edits spread across 14 genes and still get called dire wolves in headline after headline, despite being, genetically, well over 99% gray wolf. The pattern repeats because the pattern works. Name the extinct species in the title. Let the edit count do the quiet work in the background. And yet Ben Lamb, Colossal's own CEO, described the mouse publicly as bringing the company a step closer to its goal of bringing back the woolly mammoth. The engineering claim and the mammoth claim are both being made in the same sentence, and only one of them survives the gene-by-gene count. Even Beth Shapiro, the same scientist filmed drilling into that cattle jaw, has been more careful in her own language than the press release was, describing Colossal's animals as engineered proxies rather than resurrections. The person doing the science and the person selling it are not always saying the same thing. A proxy, in Shapiro's own usage, means an animal built to fill a mammoth's ecological role and resemble a mammoth in the traits that matter for that role, not a genetic replica of one. That is a coherent scientific position, and a defensible one. It is also a much smaller claim than bringing back the woolly mammoth, and the gap between those two phrases is where most of the public confusion about this entire field actually lives. Nobody at Colossal is lying when they use the bigger phrase. They are choosing the version of the truth that travels further, the one that gets picked up by CNN instead of sitting quietly in a specialist journal. So the honest version sits between the headline and the criticism. Colossal proved something real. 8 edits across 7 genes, layered into a single living mammal that survived to adulthood. What it did not prove is that a mammoth is any closer to walking around, because 5 of those 8 edits were never about the mammoth's actual genome. They were about making a mouse look like one. A working demonstration of multiplex gene editing got dressed up as a mammoth story, because a mammoth story is the one that makes the news, and a technical paper about stacking CRISPR edits mostly does not. None of that makes the woolly mouse a stunt. It makes it a data point in a much longer project, one milestone among many still to come before anything resembling a mammoth calf exists anywhere outside a press release. Watch the artificial egg built to hatch a moa next. It is the same pattern in different fur, a real piece of engineering, carrying a claim that is doing more work than the science underneath it has earned.